Hello, welcome back. We are live once again. Tonight we have a real treat. We're going to be doing a practice AP exam together. And no, not 3 hours long. We're going to do a small sample of multiple choice and then walk through a couple of guiding principles when you're working through FRQs. Hello, I'm Melanie King from the Absolute Recap. If you're not sure why you're here, we have already done a couple lives and I'll point them out to you a little bit later on if you want to go back and check those out. We have a lot to go over tonight. What I need you to do, so we're going to be working in the CED. That's the course exam description for biology, and they have these for all of the AP subjects. I have it linked for you. You can go head on over and grab that with the exam slayer. I'm going to show you how to access it once you log into the course, and so I'm going to switch screens so that you can see that a little bit easier. When you are over in Exam Slayer, this is what it looks like. You can see on the left-hand side a couple of options, and we're going to be launching inside of the simulator. The simulator that I have built for you isn't an Exam Slayer, but you can access the questions the same way. So over on the left-hand side, if you're going to be toggling down, you'll see some resources for you here. This is where I have the CED linked. You also are going to find some other videos. Here's some more practice, the one-pagers, the skills practice, and some other opportunities if you need more help. And obviously, these are where the live exam review things have been linked. So you can get this a couple different ways. If you are going to be following along with the live, there's the link. You can also access the CED PDF by clicking on this link directly. And we're going to be working through pages 200 to 216 specifically. That's where you're going to find the questions. You also can just get it directly under the resources tab by clicking the course exam description PDF. It is 224 pages. Just be mindful of the fact that the pages that you see inside this document don't all correspond to the count at the top because we start with like sub II and things like that. But you want to go to the actual page 200 on the CED document itself. And that's where we're going to be working through. Now, I personally thought it would be a lot more useful to put the questions out of PDF and into the simulator because that's what it's going to look like on exam day. And the closer we can align to exam day, especially at this point, the better the practice is going to be. So again, you want to grab the CED in order to follow along with these questions, make notes on them. You can jot notes down on the side, whatever your preference. You can scan the QR code there, and that will link you right over to it as well. You have to click on the actual product, though, in order to see the free preview link. It's not going to be on that first drop page. But I'm just curious, like, how did everyone find us? We come from a lot of different places. Maybe you are an original follower from the podcast days. This was a 2020 thing. Perhaps you are an ultimate review packet person. Maybe you stumbled upon us on YouTube. Maybe you're an AP Bio penguin and that's how you found us. Shout out to my penguins if you happen to be one. Or maybe you're from the new side, the ultimate exam slayer. But there are a lot of resources no matter where you look. So please make sure to just like find your niche. I know I'm a bio word, right? But find what works for you, especially at this stage of studying, because the worst thing that you could do, and I beg you not to do this, is to like start at September and like just gone all the way through until April and May, because you need to focus on the areas you're weakest in, not on the things and the orders in which you took things. And so that's really why you want to diversify what you use to study from. Don't just use one resource, and make sure you're practicing with different parts of your brain as you go through. So we already did units 1 through 8, and there's some practice questions in there too. So you'll find those over on YouTube. The guided notes for those as well are available. So all of that's linked up in the description of those lives, and you can still go back and watch those. They're each about an hour. I think tonight we're going to be about 45 minutes. I think it'll be a little shorter because the number of questions that we have to do is really directive. And so I think we'll wrap up relatively quickly tonight. If you have questions, please put them in the chat and we will get to as many as we possibly can. But as we work through these questions tonight, it's so important. I mean, the answer is important, I know, but why the other choices are not correct? We're going to focus there a lot because there's so many red herrings or so many terms or so many kind of like roundabout ways. I promise you, they're not trying to trick you. They, right? The College Board, they're not trying to trick you, but they do know that they need to differentiate the student who really understands and can apply the information from the student who maybe just memorized the information. And so that's why these questions are worded in the ways that they are. So we're going to walk through a lot of that tonight. All right, I'm going to make this a little bigger so that you can see it more specifically. So multiple choice tips. First off, this is a hybrid digital exam. And so that means the multiple choice will be completely digital, whereas the FRQs are going to be hybrid digital. You're going to view them on the screen, but you're going to handwrite your responses. And that has to do with the fact that 2 of the questions you have to interact with in some way. More specifically, question 2, question 5, you have to graph and you have to label or draw of a diagram or rearrange something for the reader. So until that's done digitally, and I'll be honest, I think that's not too far away, we're going to stay hybrid digital for AP Bio. All right. In the multiple choice section, you're going to find graphs, tables, charts, models. Some questions are standalone. Some questions are going to be individual stimulus based. So the prompt will look almost FRQ-like, and there'll be 4 to 5 questions about that prompt. We're going to see some of those tonight as well. Pacing, 1.5 minutes-ish per question. The ones that have a stimulus will probably take you a little bit longer. The ones that don't, you can move through a little bit more quickly. Now, you can use the Desmos calculator that's integrated into the system. But if you haven't used the calculator in the Blue Book app yet, the AP Bio exam is not a day to test it. So bring your approved scientific or graphing calculator with you. Have it adjacent to you, even if you think you may not use it. It's a little keepsake off to the side. Make sure that you do have it. Charged or bring extra batteries. That would defeat the purpose if you show up with a calculator that doesn't work. The other thing you're going to find in the app is the equations and formulas sheet. It's going to be in the upper right-hand corner. I'll show you in the simulator where ours is. It's at a really similar location, but you do have to know where to look. All of the sections in the equations and formula sheet, they're going to be condensed and collapsed to get started. So let's just say you had to find chi-square. Well, you have to know, at least without randomly clicking, that's statistics. And I need to click on the statistics drop down to get that information fastest. You can do this inside AP Central right now. So if you haven't tried that, ask your teacher to assign you some type of practice questions or practice test so you can use that within the system and get comfortable with that drop-down formulas. All right, so what strategy do I think you should use? Find the actual question. So some of these multiple choice, they're like a paragraph and a half. They have a table, they have a diagram. Just like jump to the bottom, read the actual question, then go look at the context for the question, because sometimes all the preamble isn't even needed to answer the question more directly. So not that it's like a trap, if you will, but that will cut out some time for you. Just jump to the question, the actual what line with the question mark, and then go back to the information. Captions, axis labels, diagrams, tables, those are all huge resources for you. They are kind of like freebies because they're only going to have what's labeled, what's necessary, which implies that things that have captions are really important, and you should make sure to note that in order to have this be kind of like work better for you. Now, in the actual system, you can highlight, you can cross out options, you can also use mark for review, and we're going to need to use that. I personally, I would think it would be an error if you were to not answer a question and move on. If you don't know, take a guess, mark for review, and move on. So at the end, you'll have, if you have time left, you can go and see, all right, which questions, 5, 6, 7, did I mark for review that I have to go back and look at? But if the timer runs out, you've at least given them an answer. And a guess is better than a blank. So we want to make sure we're answering every question in sequence, marking for review as we need to, and going back if some things are a little bit unclear. We'll practice doing that tonight so you can see how that works as well within the system. If you have a multiple choice tip that you've been using, whether it's something you've applied in class or something that your teacher shared, make sure to put that in the chat. I think everyone could benefit from as many examples as possible and as many strategies as possible. But we are going to practice. So once again, if you haven't yet grabbed the copy of the CED, I have it linked at the top of the chat. You can click that PDF too. You can also go and type into Google, just AP Bio CED, it'll pop up for you. We're going to be working, as I said previously, in the simulator software. So the simulator software is inside of ExamSlayer, and I took all of the questions from the CED so that we could work through them. And I want to point out a couple of areas. I'm going to make this a smidge bigger for us before we get working. A couple of areas that you can use. So here's your strikeout function that allows us to get rid of options. Here's your mark for review. So if we weren't sure, we would then be able to see, okay, number 1 has a little red flag. I know that I need to come back to that. But we haven't even read it yet, so I'm going to unclick it. Things that are different in our simulator software. Well, first off, it is not endorsed by the College Board. I do need to say that, or Blue Book. It looks real familiar, but it is not created or endorsed by them. So our timer counts up, and you can go over the time limit, but the timer is good to keep track of, because if you need to be mindful of your pacing, letting so many minutes pass for so many questions is something that you can improve on over time. The other feature that we have is the ability to show the answer. Now that's like, well, why would I want to do that? You could choose to take this without ever clicking that button. And when we get to the final screen, I'll show you what it looks like. It's going to give you answers and explanations with every question. It's also going to give you answers and explanations relative to unit, and break down what had a stimulus, what didn't, really point out some things that are useful to you. So with that in mind, we're going to get started. So if you happen to know the answer, please pop it in the chat. I will talk through some of the things I would think through with these questions, and then I will click show answers so we can look at explanations together, and make sure I hit finish test at the end so I can show you the software and what it does. Again, these are all from the 2020 AP Bio CED, all released questions from the College Board. So first off, insulin is a protein hormone that is secreted in response to elevated blood glucose levels. This is significant. Protein hormone. When insulin binds to its receptors on liver cells, on, not in, the activated receptors stimulate phosphorylation cascades that cause the translocation of glucose transporters to the plasma membrane. In your head, you're going, I have no idea what translocation of glucose transporters even means. Don't worry about it. If I had followed my own advice, I would have jumped to the question first and then gone back and looked at what insulin was doing. So what is the role of insulin in this particular example? I see a lot of people choosing A. Some are choosing B. Let's cross out some choices. It acts as a secondary messenger. Well, that's not possible because a secondary messenger would need to enter the cell. We have extracellular binding here, so that's not true. What we have binding would have to cause maybe a channel protein to open, like calcium would have to influx or something like that. Acts as a protein kinase. I don't see that happening either, because that would be a relay protein in order for that phosphorylation to happen. So that's not going to be possible. So it's a ligand or it's a receptor. Well, I see it has to be a ligand and not a receptor. And the reason, because it's the molecule that's binding. It's the molecule that's binding. So I'm going to choose A. And just for the sake of it, let's click answer and explanation. So this is the type of feedback you're going to get in these questions, meaning whether you use the ones in ExamSlayer or you use the ones in College Board, this is what we're hoping for. So it's not just why is it correct, but why are the other ones not going to make any sense? So really the tricky one in this was maybe secondary messenger. I would say sound fancier as an option, but it is a ligand. Protein hormone implies that it is going to be unable to diffuse through the membrane. If this had just said hormone, the answer would still have been ligand, but we would need it of more information because hormones could be steroid hormones, which are lipid, which can diffuse. So the protein was a really essential piece of this information. So that's question one. We're going to move on to question two. Again, I'm going to make it a little bigger for you to read. Humans have a diploid number 2n of 46. Which of the following statements best predicts the consequence if meiosis did not occur during gametogenesis? Okay. 2n is 46, diploid. That means my somatic body cell has a 46 chromosomes in homologous pairs of 23. Mitosis would maintain that number. We're talking meiosis land. So in meiosis, we have to make sure we are having the amount of genetic information because I want to produce gametes, and gametes have to then join up in fertilization to restore chromosome number. So based on that information, the question here is, what would be the consequence? I'm seeing a lot of people choosing option B. So let's go with option B. And let's look at some of these other things here that we are identifying. Gametes would get larger. Okay, option A, the gametes would get larger. That has to do with cytoplasm, and that has to do with cytokinesis, and that's not really going to be represented here. Triple, the only way D would be correct is if fertilization involved 3 organisms, and we're not seeing that happen either. So I'm going to also hit show answer here. Let's see how we did. And B is the correct answer. So when we're looking for kind of this version of a response, we want to ensure that we are honing in our vocabulary. And at times, like we did, defining some of those terms before we ever look at the answer choices, because these answer choices, well, they could convince us that they're correct. But if we take a moment to just pause, what do we know about meiosis? What's the purpose of meiosis? And then if meiosis could not happen, right? If it was blocked, what's a consequence that we would see on the other side? So great job. Question 3. All right. Ooh, too big. I'm going to mark this one for review, and I haven't even started the question. And the reason I'm going to do that is because these POU4F, these are all terrifying terms, or at least they could be. These are just examples and placeholders of a concept. You clearly didn't learn about these specific genes at any point in bio, unless your teacher did an incredible deep dive into something that predicted an AP question. Replace them. Mutations in the hot dog and hamburger genes. It doesn't matter what they're called. What matters is, can we kind of put forward information from this? So I'm going to follow my own advice. I'm going to jump to the question. Which of the following questions will help best guide the researchers toward a direct test of their proposal? This is an experimental design question. These can be a little challenging when we are kind of like bringing all of these things forward. So I'm going to give you guys just a second. And I'm going to make it bigger so we can have some answers in the chat. All right. Let's take a look here. A lot of people here choosing D. Let's choose D. But looking at why the options of the other ones aren't really great choices. A is kind of vague. C is about mice. I'm sure mice are lovely, but not relevant. This is going to be really important when we look at kind of like having this all come together. And so we go and we hit check our answer. Let's see how we did. These can be a little bit challenging, but oftentimes in these experimental design questions, we need to be mindful of the fact that it's going to have just like information that was, I don't want to say pulled from like, Animal Planet or some sort of Netflix documentary, but there's always things put in these questions that make them sound better than they are. And it's just kind of a diversion. And so we don't want to fall into any of those traps. So D is definitely the best answer here. All right. We are in the first of several stimulus questions. And so for a stimulus question, you're going to see on the exam a split screen. The left side of the split screen will have your text that you need to refer to for a series of questions. A couple of options here. You could choose to read the prompt and then the questions. That's fine. You could choose to read the questions first and then the prompt. That's probably a faster way to go through some of this. Many of these questions are only going to pull from smaller pieces of the information. So that's also something to be mindful of here. What we're looking at is phytoplankton biomass, so the amount of phytoplankton in this particular ecosystem. And they've manipulated the environment in some way to test the effect of sucrose or the effect of sucrose and phosphate together. This is something that we need to think of when we're kind of splitting these things together. The prompt here is just talking about what the researchers did, and then we'll go towards the question side. But what's interesting, and I do want to make note of this, so I'm going to hit next. The question's going to change, but the prompt is going to stay the same. And so until I get to question 8, this is something that we just need to check out for. So just a comment was made here. I was wondering if these questions were on a real AP exam. So these questions were not on a real AP exam. In fact, the College Board hasn't released AP Bio exams, multiple choice questions, since 2013. We are due. Now, in fairness to the College Board, they're actually changing the test next year. So I think this might be the year we get some questions. I'm hoping this is the year we get some questions because the ones that are released are outdated. These questions are from the CED. So if you're looking to follow along, you can go up to the top of this chat and you can grab a link. I have it linked inside both the URP and the UES. It's the free download from the College Board. All right. Which of the following claims is best supported by the data? When we look here, we're saying, okay, carbon was a limiting factor, phosphate, both carbon and phosphate, neither carbon and phosphate. So the carbon is in the sucrose. That is a unit 1 level information that you would have to know to answer this question. And so it can't be C or D. And I know that because of the shape of my graph. We're not both going up, we're not both going down. So that can't be possible. If we're looking here to whether it was carbon or whether it was phosphate, it seems that if you don't have phosphate, you're not going to be getting the response that you want. So something about the phosphate is causing an increase in the phytoplankton within this system. So let's choose B, and we are going to hit show answer, and that's exactly what we're seeing. So you add the phosphate, you have phosphate and sucrose together, and you're seeing the effect on the population growth. Okay, now here we see a calculation. You are going to be given scrap paper for the exam. So you should have pens and pencils available because you'll need them on FRQs anyways. You will have a calculator available. You could either choose to click the calculator in the platform, or you could choose to click use the calculator adjacent to you. This is a rate calculation. The rate calculation is not on the formula sheet. It is expected as a kind of base knowledge of science that you are able to do a rate calculation by pulling data off a graph. So let me make this bigger so you can see it a little bit better. Right off the bat, I'm going to take away A and D because they're such outliers to the data that they can't possibly be the options. So we're gonna cross those off. The numbers that you should have responded to is and done with your math. So you should have 6,000 divided by 4. So when we hit show answer here, that's going to give you 6,000 divided by 4 years. The correct answer here would be C. Again, this is not on the equation sheet. And so this could be something that you just want to stick in the back of your brain as a rate calculation. for you. Now, generally speaking, when you were tasked on the AP exam with pulling numbers off of a graph, they tend to give you a range of possible answers that are acceptable. Because this is multiple choice and not FRQ, obviously this was the closest answer that we would have. All right, question 6. Which of the following treatments would have been the best control treatment for the experiment? So following treatments. Remember that you have both positive and negative controls. And so it depends upon whether you are withholding the independent variable or whether you are comparing it to an expected positive result. And you're picking like a middle ground of that positive result. So if I look at my options here, we want to be mindful of choosing something that is going to allow for this to be really showing the effect of phosphorus. And so I see a mixture of people choosing A and D. So I'm going to say D is not going to be a good choice. And the reason is because small pool, making anything smaller is going to always have a greater chance to skew your data. Large sample sizes are always better. So small pool is not going to be a good option here. If we look at a different lake, that's probably not going to work either because we want to use the type of lake as a constant. Be careful. Constants and controls are not the same thing. Constants are going to be what you keep consistent from experimental group to experimental group. Like you would want to use the same species of phytoplankton in this example. That would be a constant. A control is a group that you use for comparative data. So A is a really good choice. Let's check and show our answer. And A is what we see as our response. All right, we have one more option for question 7 in our stimulus, and then we'll be about halfway done with our practice test, and we will just take a quick little breather to see where we're at. So which of the following was most likely a direct consequence of the addition of phosphate to the lake? So we need to think, what does phosphate do that might cause this increase in biomass within my lake? All right. So we do have to have a little bit of context to where phytoplankton might be within the food chain, and what these trophic levels mean. So remember that trophic levels mean troph means feeder, and so we refer to as the primary trophic level, secondary, tertiary. As we go higher up, available energy tends to decrease as you go higher up. But we also can find that the number of organisms also decreases. And so I see a mixture of C and D mostly in the chat. All right, I want to talk about choice C. The amount of energy available to producers in the lake increased. Producers capture energy from sunlight, or chemosynthesis too. The only way for C to be true, and I can see some of my own students in the chat who chose C, the only way for C to be true would be increasing available sunlight. And so C cannot be the possibility for answer. So let's choose D and click show answer. And D is correct. So be careful. Just because we're talking about producers doesn't mean that that's going to be the response that we're looking for. All right, we're moving on from the stimulus. At least the stimulus sets. Okay, at least the stimulus sets. So here's an example of a question that does have a stimulus, but the stimulus is not going to be carried through multiple questions. All right. So pH graphs with activity of enzymes, classic. You might also see this with temperature or salinity or what have you. Moral of the story is enzymes have Goldilocks zones. They only function best in certain types of environments. You go outside the Goldilocks zone, things start to happen. I say things intentionally because that's part of the answer to this question. So I'm going to remove myself, make this a little bit bigger for you guys to pop in the chat. All right, we're almost exclusively all choosing D here, so I'm not going to linger on this question too much, but what I do want to think on is what denaturing means. So denaturing is going to cause the conformational, the 3D shape of my protein to be altered. And that can happen sometimes in a reversible way and sometimes in a non-reversible way. We are not breaking peptide bonds. So the primary structure of my protein is going to remain intact. I am, however, losing my conformational shape. If my enzyme does not have the right shape, it will not have an effective function. And so that's why we end up with this peak in our graph for this type of question. All right. There is some tables to digest here. So I'm going to leave the screen, turn off my mic for about 30 seconds to give us some time to think. All right. So almost exclusively choosing D in the chat, but I do want to talk just a little bit about choice A. This particular diagram has absolutely no reference to time, okay? The fact that they're labeled A, B, C, and D, and it's sequential, and that they're listed left to right in order means absolutely nothing. Okay, it just happens to be the way that this particular scientist put their data together. If this was a different scientist, it would have been shuffled around. It wouldn't have had the same plus minus locations. The answer would still have been the same. And that's important to be mindful of as we go through this. And so if we're looking here, the fact that everybody has chlorophyll A is going to imply common ancestor. Now, this is a character chart, and you could use this particular character chart to draw a phylogenetic tree to show relatedness. That would be a linkage between these two. That's the type of question you might see in an FRQ as you were going through the concepts. Yeah, the other choices, they sounded real wordy, and sometimes that can be a clue in and of itself that it's probably not the answer. All right. You might have done this lab in class. It's kind of a classic, maybe biolab or AP biolab, just depending. So we're looking here at a wet mount. We're adding salt solution, NaCl. And then they're looking into what happened to these cells. These are plant cells. I can see my cell walls. It's also showing you the red onion. We're looking for an example. So I think it would just be good practice to use some good terminology here before we jump into the answers. We have added a hypertonic solution around the cells because the salt concentration is greater than it had previously been. So in that circumstance, if I have a hypertonic solution outside, I am hypotonic inside, and hypotonic is going to have water flow from the inside of the cell to the outside of the cell. The salt, if it could move, right? It's ionizing most likely, but if it were to move through some type of channel protein, it would also follow its own concentration gradient, high to low. Salt is really small. Probably not going to see a large effect on this particular cell. So if we look at our options here, degradation of the DNA in the nuclei, we have no information about that. That's an assumption. So let's cross that one out. Lysis of chloroplasts in the cells. It's poor chloroplast. So lysis refers to cutting or chopping into pieces. So we see that with like hydrolysis as an example. C looks to be what I literally just described. So let's choose C. And then D, the movement of NaCl from the solution into the cytoplasm of the cells. Now, had this been the movement of the solute as part of your response, and that was such a significant movement that the cells did swell, you would then have a filling of the cell. And so the question is like, why are you seeing what you're seeing in the slides? And the movement of salt isn't going to impact the visual that these cells are showing you like you would get in, with the withdrawal of water. Just as a curiosity, does anyone know what this condition is called when you have plant cells that look like this? It has a very specific term. Just curious. I highly doubt it's going to be an answer on the exam, but it might be in a question. When you have water leaving a cell and you have bringing in when you have the plasma membrane that's been shrunk down, we are going to call, I see a vote for flaccid. That would be true if it was an animal cell, but it is not an animal cell. It is a plant cell. And so we do have a different terminology here. So we're looking for plasmolysis or plasmolyzed as our response of this answer. So we'll give a shout out to the one in the chat who did just get the correct answer. Well done, my friend. All right. All right. So I'm going to pause. We're about halfway through and I want to answer this question. On the actual exam, will we be able to revisit questions? I read that we weren't going to be able to. Yes, you can. It's called Mark for Review. It is something that you will have. So that's what this is showing up at the top. We do have this feature, excuse me, at the top. And then you could check on the bottom to show which questions have been marked and which questions have not been marked. All right. So we are at 9:38 and we are on question 11. We have 15 questions to go through tonight. So I'm going to expand the screen so you can see this a little bit better. I think. Ooh, there we go. And give you guys some time to think. And I will be right back. All right. We are all over the place in the chat as an answer to this. Before we get to this answer, I want to address one of the other questions I just got. Has the CED changed since 2020? No, it's changing next year. But there has been a change in question format, and that is due to the hybrid digital, which is what you'll experience this year, multiple choice, all digital, FRQs viewed on screen, but still handwritten. And there's been a change in the structure of the FRQs, meaning, and this was last year, but a lot of the sample FRQs you're going to see in AP Classroom, you're going to see on the College Board website, have the old structure. And so the FRQs this year, they have the questions kind of like sprinkled within the prompt. And so you're going to have like a paragraph and then part A, subpart I, and then a table, and then AII, and then a graph, and then part B. So it's going to help allow you to predict and have a much greater association for how much information do you actually need to answer the question? It's not going to be everything. And so that's important to be aware of, because you don't have to have all of the information from the FRQ to answer every subpart. And this is going to help guide you in that. So you'll see it broken down a little bit better. All right, let's look at this question 11, and see where we're at. We have people in the chat talking about exons and introns, right? So introns are interrupting. Exons are expressed. Okay, so that's the phrasing you can use in order to kind of have this kind of terminology. So we can do what is referred to as alternative splicing in order to ensure that we have the actual product we need from the same genes. You get this differentiation here. And so with that kind of, we'll call it tip, a lot of us are choosing B. Choice C, just so you guys are aware, so the GTP cap, that's the 5' side. What that does is it helps the transcript exit the nucleus and bind to the ribosome. So it's really useful, but that's not, although it's a true piece of information, sort of, it's not going to be the answer to this particular question. So let's hit show answer just to double check. And we do get that answer here. Yeah, sounds good. All right. Closing it on the end, and then final tips coming up. GTP cap and guanine cap are the same thing. So it's the opposite end. On the other side, we'd have a poly A tail. All right, I'm confused. Why are the exons spliced? Exons are expressed. They're not spliced out. Introns are removed. Introns are interrupting sequences. They might be genes in other contexts, but sometimes they're just garbage, and that's not what we're going to see. All right, here we have a protein. If you were in my live the other night, we actually did this question as an example from this unit. But protein that has been put in water and it clumps up. Remember that the amino acid structure that we have as the monomer for proteins has a carboxyl group on one side, an amino group on the other, a hydrogen, and then fill in the blank R group. That could be that could be just depending upon polar, non-polar. It's going to influence how we fold as a protein. It's also going to influence what shape that chain takes and how it fits into membranes. So which of the following best explains the location of the amino acids in the folded protein? We're looking for something here that's going to really reference different components of how this is folding. So polar R groups or non-polar R groups and how they're going to be interacting. Polar R groups on the surface form ionic bonds with charge. That's going to be not possible because that has to be a transfer of an electron. So we're going to cross that out as a choice. Polar R groups are too bulky to fit in the middle of the protein, are pushed towards the protein surface. No, that doesn't make much sense either. Non-polar R groups cannot form hydrogen bonds with water and are pushed to the middle of the protein. Or non-polar R groups from different parts of the protein form covalent bonds with each other to maintain the protein structure. That's just really true of all R groups. It doesn't really explain the pattern at all. So I think C is our best choice here. Let's hit show answer just to make sure. And we are on the right track. Good stuff, guys. All right, question 12 is done. Let's move on to 13, which I've clearly already answered. Well, that's all right. Maybe that's the right answer. I'll have to re-click it. All right. Looking at a fly, and we are working through here with specifically a temporal isolation. So temporal isolation is referring to the fact that these particular organisms are going to mate at different times and what that will impact moving forward. So if they're going to mate at different times, that's going to impact some things about their ability to survive as a species. Something about insecticide. I mean, insecticide is not even mentioned. So we're going to say A is not correct B, the single fly species will evolve into two distinct species because of the lack of gene flow. That seems promising, but let's check the others. The ability to survive on a diet of two different fruits will help the flies learn to eat. Okay, I'm going to stop. The flies aren't learning anything. Please be careful about your terminology when you're talking about organisms wanting to get things, becoming things, hoping to get traits. It's not going to be the answer. The flies that eat the hawthorn fruit will lay some of their eggs on the earlier ripening apples to minimize competition among the larvae. We have like a fight to the death once born moment. I think there's enough apples to go around. There's definitely more apples than the larvae could ever consume. B, we're going to stick with B as our answer. And that's exactly what we see happening here. Timing matters substantially. All right, 2 more questions for this live. Here's another example of a stimulus. On the left and the right is our question. We are not going to have to scroll through a stimulus, and it doesn't apply to more than one question. Here we're forming a platelet plug, and I'm going to jump to the question, directly to the question before I go back. Which of the following best explains the feedback mechanism illustrated in figure 1? Scanning my choices, we have positive and we have negative. As a reminder, positive is going to move an organism and a circumstance further away from homeostasis. They are temporary circumstances. They are also going to be represented far less in biology. Negative feedback is going to be situations and responses that return organisms to homeostasis, to a set point. They put things back in regulation. So, what do we think is our choices here? What did I do? What did I do? Somehow I managed to make my screen go black, but I think I fixed it now. We should be all set. All right, looking at the options here, we should be pretty aware that plugging up holes inside of your body shouldn't be a regular occurrence. So from that, I'm going to say negative feedback, probably not my choices. Let's look at the positive feedback options. Because I'm amplifying the circumstance, because I am moving away and I'm having my action, my released chemicals are actually causing more platelets to be released. A is our correct answer. Looking at the options here. All right. I just want to say I do totally appreciate you guys helping each other in the chat, especially when I can't get to all the questions, clicking through all my boxes. So if you can help some guidance, and I saw use your arrows, great tip, especially when looking at unfamiliar diagrams. All right, last question, and we will finish our test and look at our results. Oxygenation. It is estimated that oxygen production first evolved in photosynthetic prokaryotes approximately 2.7 billion years ago. That's cool. Not relevant to the question, really. Question, which of the following best supports the claim that photosynthetic prokaryotes were responsible for the oxygen in Earth's atmosphere? Okay. Please look at option B and option D. So I'm going to cross out A and C as choices. That is such a clue for you that one of these is probably correct because they are nearly the same answer, but with just different justifications. So whether it's the light reaction or the Calvin cycle, water's being split. Okay, now thinking back to what you remember about photosynthesis, we're going to lean in here to our light reactions. And just checking for show answer. All right. Great job. I'm going to hit finish test. Now, it's letting me know that I have unanswered questions. I'm going to hit submit anyways. Let's see what happened. It's very possible that you know what? Let's check. I'm going to hit my mark for review. I did not answer number 5. Oh no. Number 5. I didn't answer number 5. Number 5 was C. Okay. Now I'm going to hit finish test. And we should see our raw score at the end. Now, I cheated a little bit and was looking at the show answers. We were also talking through it. This is what your results would look like if you use the AP exam simulator inside the exam slayer. It's going to tell you questions that had units. It's going to tell you things that had stimulus. It's also then going to tell you how much time you spent on every single question. So we were talking through them. So 3 minutes and 27 seconds is not great. But it's going to show you what your pacing was as you go through all these questions. So remember that ideally we would have pacing that was 90 seconds per question. This would be a really great way to check, am I going to be effective in my pacing? And so just depending upon like where you are with your studying, that's a valuable piece of information. It's also going to tell you where you were with the different units. So like, how did you do within 5, 6, and 7? Which questions were those? Jump back to those questions. Had we selected wrong answers, we would see red on this screen, but since that wasn't the goal of tonight, I think it would have been an unfortunate thing had I gone and done wrong answers with you intentionally. I definitely think that that was not going to be functional. So I see people talking in the chat like, how do I get the exam slayer? How do I get the ultimate review packet? You could start here. That would be a great thing. I have the comment at the top. I also have in the description links to get there. But even if you don't get it at all, I still want you to sign up for this free sample. And here's why. Every teacher that has an ultimate review packet and an exam slayer took 10 of their best questions, myself included. We put them in a free course for you. So if you're taking 4 to 5 APs, that's 40, 50 free practice questions, like in the exam simulator that you get to try, feedback that you get to get. So definitely sign up for the free preview and sign up for this free course. This gets you logged in to all those sample questions. And so if you do nothing else to practice, and this is like a last ditch, I just need to make sure I do some extra questions, make sure you log into this. So this is true. So AP Calc, AP Chem, Gov, Macro, Micro, whatever you happen to be taking in addition to AP Bio, you can do that. So I do have the questions that I'm referencing myself. I do have them inside of Exam Slayer. And so I have them. I can just show them to you if I pop this up real quick. Click on the right thing. It's half the battle of doing these lives is clicking on the right thing. So here we go. Add to stage and click over. No, I'm not on the right thing. We're gonna abandon that strategy. But we have one more live left. And this particular live that we're going to do, I will put a notice here on YouTube, but we are going to be going live inside the URP and the UES. So I will pop on YouTube. I'll say hi. I'll remind you that that's where the links are. If you happen to already have the Ultimate Review Packet, if you happen to have Exam Slayer, I thank you. And this is my thank you back to you. So our last live, which is going to do strategy, pitfalls, like a last-ditch effort, probably about an hour and a half long. It's going to be on Saturday night. And it's going to be as a thank you to all of those who have been subscribed to my products. And I wish you all the best of luck. If you did not see units 1 through 8, we have those already linked for you. All those guided notes are free to download, and all the links are there for you as well. So I appreciate you. Good luck on all of your exams. AP Bio is 8 a.m. on the first day. I can't believe it. Have a great night, everybody.